Hello, my name is Tristen Eddy, and I am of the Diné Nation, which resides in the present-day Southwest. Firstly, I would like to acknowledge the indigenous groups whose land present-day Xavier University and New Orleans resides on, including but not limited to the Chitimacha tribe, the Chushata tribe, the Geneban Choctaw Indians, and the Tunica-Biloxi Indian tribe. Today, I'll be presenting on my research project titled Manipulating Murine Apolipoprotein C III to Combat Cardiovascular Diseases. To provide some background, cardiovascular diseases, or CVDs, are the leading causes of death worldwide, with an estimated 17.9 million deaths in 2019. This is no different for American Indian and Alaska Native populations, where they were 50% more likely to be diagnosed with coronary heart disease than their white counterparts in 2018. This graphic illustrates that incidence. Also, quick note, CVD is an umbrella term for many diseases of the heart and blood vessels, which can include coronary heart disease because it is a type of heart disease, but a coronary heart disease doesn't necessarily include CVDs. There are numerous risk factors for CVDs, some of which can be population-specific or even environmentally specific, but some of the important ones I've highlighted here are diabetes, low HDL cholesterol levels, and the significant one we'll focus on today, high triglyceride levels or TG levels. The TG levels are really what provide the basis for what we'll be discussing because of its relationship to the incidence of CVD. It has been identified as a determinate cardiovascular risk in some sources. In order to combat CVD incidence, Apolipoprotein C III, or ApoC3, has been targeted as a source for therapeutics because it is a mediator for TG metabolism, and previous studies have shown some promise with reducing antibody production, but was done with human ApoC3 and murine samples. This study will focus on the murine-murine interactions of ApoC3 and murine samples. In tying all of this together, ApoC3 will be attached to a virus-like particle so that it can be presented to the host immune system as an antigen, and then further exposure of the antigen or ApoC3 to the immune system will induce an immune response that will hopefully lower the presence of ApoC3 and in turn TG levels. The structure of ApoC3 itself is a 79 amino acid glycoprotein whose native structure is primarily alpha helical. The similarities between the human and mouse ApoC3 provide the foundation for this work. There are 4 total alpha helical structures in murine ApoC3, which we converted into 6 short peptide sequences. The significance of the alpha helical structures has to do with the antibody formation of the host. We postulate that the alpha helices are going to be essential in the host's production of antibodies. So controlling the helicity of the short peptides will be done via macrocyclic lactam formation. In the methods, we focus on the biochemical manipulation and the synthesis and characterization of the peptides. The macrocyclic lactam formation is a Diels-Alder reaction. This reaction is done on resin after the special protecting groups are removed. In this specific peptide, the MMT of the lysine and the ODMAB of the aspartic acid is removed to expose the amino acids. Then, using BOP and DiPA, the bond will form between the amino acids and manipulate the whole of the peptide into an alpha helix. The synthesis of each peptide is done through solid-phase peptide synthesis. The synthesis of each peptide is done on a resin ball, and then each amino acid is added from C to N until the peptide is complete. The characterization methods are important procedures that allow us to confirm different traits, if you will, about the peptide. The MALDI or ESI are procedures that measure the mass of the peptide, which will allow us to confirm a successfully synthesized peptide. To the left is an example of an ESI that confirms the correct mass of a peptide showing success. The HDL-C is used to see the purity of the peptide and a purification method in itself. On the right is an HDL-C graph that shows the purity of the peptide, so the area under the graph allows us to confirm that the purity of the peptide is over 90%. CD will allow us to confirm secondary structure, and we can compare pre and post reaction. Lastly, SDS and agarose gels are going to be used in the bio portion of the project, which is the virus-like particle conjugations. So where am I at now? I have chosen 3 variants to work with due to time constraints, and each variant is a wild-type sequence and a modified sequence reflecting our special protecting groups that we will be using. As is consistent with science, I've run into some unforeseen issues with lab machinery, so I was stalled for a bit, but will be weighing my peptides to confirm the structures. Some future steps in the next month will be to conduct the actual chemical manipulation reactions, some further characterization, VLP conjugations, and future steps will be animal studies. Furthermore, I would like to acknowledge Dr. Naomi Lee for being a mentor I could never have wished for, but I've been blessed to know, and thank you so much to the Strong Heart Study and the Partnership for Native American Cancer Prevention for the endless support. Thank you so much, and have a great day.